This lecture covers information about the cell cycle and the stages of mitosis. To understand cell division, we have to understand the cell cycle, which is the steps through which the cell goes over time before it divides. So the most of the cell cycle is actually a non-dividing state called interphase. And then there's a small part of the cell cycle for many cells when it gets called into division, and we call that the mitotic phase, or M phase. Within interphase, there are three parts. There's the G1 part, the S, and the G2. Within the mitotic phase, there are also two parts. We have mitosis, and then our little last part here, which overlaps with mitosis actually, is called cytokinesis. So what are these parts of the cell cycle? So let's talk about the stages of interphase. So interphase itself is not dormant. It's all the normal cell processes are occurring. And particularly when it's G1, the growth is happening. That's G1's main activity. And this makes sense that it would be a growth phase because this is right after division. The mitotic phase is the division phase, and so the cells need to grow back up again. The cell cycle does go around. I've drawn it in a clockwise direction here. Okay, those are arrows. If the cell, so then the cell goes through G1, and if the cell receives a signal, it gets past what we call the G1 checkpoint. So the G1 checkpoint, any checkpoint, means that you need external signals in order, or internal signals, in order to go to the next phase. So the G1 checkpoint particularly needs things like growth factors in the environment that tell the cell, okay, it's time to start going into the process that will lead to division. It also is going to need nutrients in the environment. So those are two of the many things that the G1 checkpoint has to have in order to get past. If the G1 checkpoint does not get passed, the cell can be moved into a phase called G0. Again, like all interphase stages, it's not inactive, it's just the non-dividing state. For long-term, the long-term non-dividing state. Most adult body cells are in G0. A lot of them can be called back out of G0, back into G1, except for things like nerves, neurons, I should say, because that's the cell, and muscle cells in adult humans particularly. Pretty much stay in G0 the majority of their existence, which is why when you damage neurons or muscle cells, it's so difficult to recover from that because the cells don't get pulled back into the division cycle. They just stay in G0 still, with the damage not being replaced. So the cell does all its normal functions in G0. All the normal chemical reactions, making the ATP, making food if it's photosynthesizing, building all its proteins, all that kind of stuff. It moves around if that's the cell type, cytoskeletal dynamics. And so that's all going to be in the G0. Then we get to S phase. So S stands for synthesis. I guess I should mention G stands for gap, gap 1. But I think of G means growth. So synthesis is meaning DNA replication. That's what's getting synthesized in S phase. So you'll notice the DNA replication is actually in interphase. Sometimes people are confused about that, so the DNA gets synthesized in interphase. Then we get to G2. And G2 is a phase of preparing for cell division. A little more growth. It duplicates the centrosomes with the centrioles if it's an animal cell. And then it gets to the part here, which is called the G2 checkpoint. And this is another one where the cell needs the correct combination of nutrients and signals in order to go into the actual division called mitosis. There's another checkpoint in the middle of mitosis, so we can draw that in here. We'll draw the stages of mitosis in a minute in a zoom-in picture, but this is the M checkpoint. So we have it all in the same picture. And that's going to be a transitional checkpoint within mitosis and internal signals there. So now let's look at a zoomed in picture of the mitotic phase. Alright, from our last picture, here's our mitotic phase. And we're just going to make it bigger. Remember, this is going from right to left because of our pie we drew on the last page. Remember, this is our cell cycle. And we're in the M right now. Okay, so in the M, the way you can remember the stages is pee-pee-mat, like the little training pad the dogs go pee-pee on when you're training them, housebreaking them. So it's a pee-pee-mat, and this is going to be drawn from right to left in this case because that's the stages order. So the first stage, the first P, is called prophase. Pro means before, so it's the very first phase. Now we're in the mitotic phase at this point. So that's the first P. Then we get to the next P. Now it's another pro, but this time it's pro metaphase. And that actually means before metaphase. So not surprisingly, the next phase is metaphase. Metaphase, you're going to remember the M for metaphase is going to equal middle because the chromosomes are going to be in the middle. The stage after that is called anaphase. With anaphase, you'll remember A is for away. That's when the chromosomes move away from each other. And we have our last phase, the PPMAT, is telophase or telophase. And telo means something like ends, like a telephone. You're on two ends of a line. So that's the last phase. And this, the blue, is your mitosis. The checkpoint is between metaphase and anaphase. So the cells actually have to be, the chromosomes. The chromosomes have to be lined up in the middle before they can get pulled away from each other. So the M checkpoint is an internal signal there. And then the last part of the mitotic phase is overlapping telophase a little bit. The end of that. And we call that cytokinesis. And so cytokinesis is the actual division of the cytoplasm, which is the cytosol and the organelles. So it's the cell actually physically divides. Mitosis is actually just the division of the nucleus. And this whole thing together is all called the M or mitotic phase. Let's look at each of the parts of the mitotic phase in turn. Draw little pictures. So this, remember, is your P mat. and then your overlap of cytokinesis. Cytokinesis is not part of mitosis. Neither is interphase that happens prior to mitosis. So we need to start, though, in G1. So in G1, we'll draw a cell, and the cell is going to have one centrosome. Okay, so this is a centrosome. The centrosomes are the collection of proteins, and in animal cells, these two centrioles, they're at a right angle to each other, so those are in the centrosome. So there's one of them. We have a nuclear envelope in the cell, and then we have DNA inside the cell, which is all uncoiled. So this is the nuclear envelope. We have decondensed DNA. And it's all the chromosomes. And each of the chromosomes, if we looked at them, each of them would look like this. So it's one chromosome. We could also draw it like that. So those are the 3 chromosomes. So at this point in the cell, we have 3 chromosomes. That's G1. Then what happens? We go to S phase. So in S phase, S is for synthesis of DNA. We still have a nuclear envelope with our DNA inside. It's still decondensed. We still have one centrosome. So it looks kind of pretty much the same, except for if we looked inside at the DNA, now the DNA looks like this. Or if we drew it, something along the lines of that. So what does that mean? This means it's duplicated DNA. So there's 2 what we call sister chromatids, which should be identical. So for every one chromosome, it used to be one single copy of DNA, so this is one helix. And now it's 2 helices. They're attached to each other. They're in one cell. So we call these sister chromatids. And actually, this is all still one chromosome, though. So we start with one chromosome. In the beginning, that's G1, and then by the end of S phase, we still have one chromosome. It's just duplicated now. Okay. G2 is going to look the same way because it's and actually all the way through M for metaphase is going to all look the same way. So that's going to move onward. So we've duplicated our DNA in S phase. So now what happens? Now we're going to go into G2. Still have a nuclear envelope. Still have decondensed DNA. We're still in interphase, by the way. This is all interphase. Our DNA is replicated still, duplicated. And now we're going to duplicate the centrosome. So now we have 2 of those. Still in these, we have the number of chromosomes equaling 3. It's just twice the amount of DNA, but each one counts as 1 chromosome still. Okay, after G2, that's when we go, if we get the signal, we go into actual mitosis. So now we are in big mitosis. That was all interphase before. Now we're in mitosis. So we still have our one cell. The first phase of mitosis is called the prophase. Prophase has a nuclear envelope. the centrosomes are present, they're going to start moving away from each other to opposite sides of the cell, opposite poles. And as they do that, they're actually going to make these structures called spindle fibers, and they are actually made of, I'll draw a close-up, of microtubules, and they originate and grow from the centrosome. So these are spindle fibers made from microtubules. Those are those contractile proteins, and they are going to help pull the chromosomes apart. So they're going to start to form, and the centrosomes are moving around to opposite poles of the cell. The DNA is starting to condense inside of the nuclear envelope, which means that we're starting to be able to see chromosomes. So I'll draw our chromosomes. They're not in any kind of particular place. There are just all three of them there. And that is our prophase. Next, we get to what is called pro-metaphase, which is characterized when the centrosomes are on opposite sides of the cell. And the spindle has formed. What's happening also is that your nuclear envelope is broken up so that the spindle is able to get into, reach the chromosomes inside. And then our chromosomes that are condensed are going to be getting attached to spindle fibers. So the spindle fibers kind of grow and shrink and grow and shrink until they connect with this middle part, or a part on the chromosome called the kinetochore. So if we look again at our chromosomes, there's this region here we call the centromere, the region. It's the middle part of the chromosome. Actually, it's the skinny part of the chromosome. Some chromosomes have their centromeres near their top. Some chromosomes have it in the middle. So they could have chromosomes like this, and this would be the centromere. So that's the region, and at the centromere there are complexes of proteins called the kinetochore. So the kinetochore is where the microtubules come in and they attach to. Still being studied, not quite clear how everything works in it, but it's definitely a protein complex with a lot of proteins. And so these spindles are coming in and out and in and out, and eventually they'll connect to the kinetochore and bind there. The main goal is to get spindles coming in on either side, the other and the other sister chromatid, and that's going to basically orient the chromosomes to where they're getting ready to be pulled apart towards the centrosomes. Makes sense to have them come in from the side, because then the pulling action just takes them back. We still have 3 chromosomes total. Moving from there, we go to metaphase. Remember, we said metaphase means middle. So if we look at our cell now, no nuclear envelope anymore. We do have our centrosomes on opposite sides still. We have our spindle that at this point has hooked up to our chromosomes at their kinetochores. At least some of the spindle fibers have. So, and pulled them. So the spindles are very mechanical. They actually can pull back and forth until they tug the chromosomes to the middle of the cell, because there'll be equal tension on both sides of the chromosome. But you can see at this point we have microtubules attached to the chromosomes, and the chromosomes are lined up single file. In the middle of the cell, or the equator of the cell. It's not a real plate or anything like that, metaphase plate. It just happens to be in the middle. Remember, this is in 3 dimensions. It would be in the middle of the cell in all dimensions. So that is metaphase. Now the chromosomes are lined up, and when the tension is equal on both sides of the chromosome, there is a signal that tells the cell that it's ready to pull apart, and then it will pass that checkpoint and go to anaphase. Anaphase is away. Again, there's no nuclear envelope or anything. There are the centrosomes, and there are the spindle fibers, but now the spindle fibers are shortening, and the cell, I can make this more pronounced, is actually elongated. So what's happening, though, is the chromosomes have been split, the sister chromatids, and now they're being pulled in by the microtubules of the spindle. Okay, so they're moving away from each other towards the poles. And what this is going to do is this is going to mean that they're far away, so when we split the cell in half, it's most likely to get this part in one cell and this part in another cell, which is what we want. In metaphase, we had 3 chromosomes total, and now we have 6. Each of the separated sister chromatids counts as its own chromosome now, so you saw 3 on that side and 3 on this side. Then the last phase we have is telophase, the end phase, and that one, draw an early telophase here. We have our chromosomes starting to decondense again on both sides of the cell, and a nuclear envelope is starting to reform around each of those sets of chromosomes. The microtubules have gone away. We still have the centrosome. It'll stay outside the nucleus, but the microtubules are pretty much gone. We can see later telophase, which is overlapping with cytokinesis. This is an animal cell. As a plant cell, I would have been drawing a little square, but draw a plant cell. So this last part is going to be the late telophase, as well as cytokinesis. Remember, that's part of the mitotic phase, but not mitosis. Here we have our condensed DNA, or decondensed DNA, our nuclear envelope basically fully formed. And then we have the division happening. That's the cytokinesis. Now you can see we've already done mitosis because we've divided the nuclei, but now we need to divide the cell completely. So this is in animal cells, cells without cell walls. This is called a cleavage furrow. This would also be cells like yeast don't have cell walls. They make cleavage furrows, budding yeast at least. And then this is like a cell. This is a cell-walled organism like a plant. Plants have what we call a cell plate that forms. Cell plate. Remember, all the organelles are all in these cells. So the cell plate forms from little vesicles from the Golgi apparatus that come on cytoskeletal elements to the middle of the cell. They're containing proteins, enzymes that make cellulose, and cellulose gets deposited in the middle of the cell. And then eventually, the cells would become 2 separate entities, and you'd end up with 2 cells, and they should look like the original parent cell, including the number of chromosomes in each new nucleus at 3. I should point out that in early telophase and later telophase, both of the new forming nuclei have 3 chromosomes, but overall, since these aren't split yet, the entire cell will have 6 chromosomes total. And then 1 centrosome hanging out. And the cell wall just builds based off that cell plate, and it'll just create a cell wall there. Now you'll have 2 plant cells that are half the size of what the parent cell was, and they'll just go through G1 and grow and become the size of the original parent cells with 3 chromosomes in each new nucleus. So that is PPMAT. prophase, prometaphase, metaphase, anaphase, telophase, and overlapping with that, the phase of cytokinesis, all making up the mitotic phase of the cell cycle. Here's a beautiful picture of the process we just talked about. You can see the cell starts with one nucleus, the chromosomes condense, they line up in the middle, they pull away from each other, and then they decondense into two nuclei now. So we've just seen the process all color-coded in sequence of a mitotic division. Organisms need to undergo cell division for a number of reasons. Three big ones are the growth of the organism. So multicellular organisms all start as fertilized eggs, which are single cells, and those need to be able to divide to make identical copies of the cells in order to make a multicellular organism. We also need maintenance, so when cells are injured or they just slough off, really. We also need maintenance, so when cells life cycle is done, so cells only live for so long, they're going to slough off and you need to replace those cells. And then we also have repair. For example, when you get a cut, you need to replace the cells that have been removed, and that's done through cell division as well. The types of division we're looking at so far all result in genetically identical daughter cells. That's as long as the DNA replication, the S phase of the cell cycle, properly copies to DNA. And that makes sense that these would be body cells that were doing this, because you have a skin cell, you want to make more skin cells, you want all those skin cells to look just like the parent cell initially. So the parent cells and the daughter cells should all be the same. And in eukaryotes, we'd use mitosis to do that process. You do need to know some definitions. For example, genome is all the DNA inside a cell's nuclear region. So it's all the chromosomes, all accounted for, the genes and the non-coding parts. That's all the DNA is the genome. Remember, prokaryotes have a single circular chromosome. That's their whole genome. Eukaryotic cells, it's all the linear chromosomes all together. Chromosomes themselves are DNA molecules that are wrapped around proteins. Chromatin is the actual DNA protein complex, and that's going to condense during cell division. You can start to see the individual chromosomes now because they are tightening, they're getting wound up around those proteins much more tightly, so that when the cell divides, the chromosomes don't get all tangled in each other and break as the cell's pulling them apart. Prokaryotes can't undergo mitosis because they don't have a nucleus, and that's the definition of mitosis. And that's the definition of mitosis, the division of the nucleus. So what ends up happening, they do a process called binary fission. You can see here is a bacterial cell, a prokaryotic single circular chromosome. Part of that chromosome is attached to the cell wall at this stage. The chromosome is going to replicate, so now we have two chromosomes, the other one's attached to the other side of the cell wall. And this cell elongates and starts to build a cell wall in between the two full genomes, and eventually that splits in half, and now you have two new prokaryotic cells, in this case bacteria, when you used to just have one. So these are the two daughter cells, the daughter bacteria, and each has a complete genome that's identical to the parent genome. There are some interesting examples of variations on cell division. So we just saw binary fission. We've seen mitosis, but there's some sort of in-between kind. So, for example, in dinoflagellates, which are a type of protist group, the nuclear envelope, since it's a protist, it's a eukaryote, but it stays intact. But the microtubules go through these openings in it, and that's what helps pull the chromosomes apart. So it's sort of a variation in between breaking down the nuclear envelope completely and not even having a nuclear envelope at all. We also see diatoms, another type of protist, and some yeasts, which are a type of fungus. They also maintain their nuclear envelope through division. You can see all the microtubules form inside of that nuclear envelope and do the cell division there. And most eukaryotes, though, the nuclear envelope fragments, and that way the chromosomes can get as far apart from each other as possible, so that it's higher probability that when the cell divides into two parts, each part will only get half the amount of chromosomes. Here's an example of a plant cell undergoing mitosis. So you'd need to be able to identify these stages if I gave you pictures of them, real pictures or sketches. So here's our prophase. So our prophase is when we see the nucleolus still, the chromosomes are just starting to condense. Prometaphase, the chromosomes are much more condensed, but they're still contained in relatively in the nuclear area. It's starting to break up now, though. Metaphase, the chromosomes are lined up on the middle. These are the arms of the chromosome. So remember, it lines up by the centromere part. So those are sticking off to the sides. This is very early anaphase, but anaphase is when the chromosomes pull away from each other. Later on, we'll see them further apart. And here's telophase. You can tell because the chromosomes are decondensing. You can't see them as well now. And you're getting this nuclear envelope forming around each of those condensed chromosomes. And then we get to telophase, where the chromosomes are decondensing. You can tell that because you can't distinguish them very well anymore. And the nuclear envelope's reforming. You can see that boundary forming around those decondensed chromosomes. This is a cell that's a plant. And here is the cell plate forming. And eventually that cell plate will hook up on the sides, make a new cell wall. And then you'll have, where you had one cell, you'll have two daughter cells, each half the size. Each of those daughters will go through G1, grow up into the same size as a parent in many cases, and then they can divide too if they get the right signals. You'd also want to know what mitosis and cytokinesis looks like in animals. You'll also want to know what you'll also want to be able to identify pictures of mitosis and cytokinesis in animals. These are some nice images of cytokinesis in animals versus plants. So in plants, we just saw that there's the cell wall. So the cell wall here is forming from these little vesicles from the Golgi, bring the enzymes that lay down the cellulose. The cellulose makes the cell plate. Cell plate eventually forms the cell wall. You can see here's the nuclei. Here's the cleavage furrow. It's really made from actin contracting. So actin filaments contract like a drawstring bag, and it basically pinches the two cells apart. So it pinches that one cell into two cells. And obviously animals can do this, but plants can't because the plants have that cell wall. And so it's too rigid to pinch. And even if they just pinch the cytoplasm or the cell membrane in there, you'd still have two cells within one cell wall, which is a problem. So they build a new cell wall if you're a plant. You just pinch in half if you are an animal. We talked about the cell cycle checkpoints when we drew out the stages of the cell cycle. And here are some checkpoint factors. So these are factors that keep the cells from moving beyond the checkpoint when the conditions are not appropriate. So one thing is called density-dependent inhibition. So what density-dependent inhibition means is that when you're a cell and you contact cells next to you, there are signals between the two cells because of receptors on the membrane that tell the cells to stop dividing. So basically they keep the cell from going through the G1 checkpoint. So the cell will stay in G0 until the cells are removed, and now there's a gap, or until any cells are removed and now there's a hole. Those cells are not getting the signal anymore to stop dividing, so they get past the G1 checkpoint and do a phase or a round of two of mitosis, and then they fill that hole back in, and now they're touching again, so they stop dividing. That's density-dependent inhibition. Another signal that cells have to have in order to divide is anchorage dependence. So that just means that the cells need to be anchored to a substratum in order to divide. They can't be floating around in the body. And so, like when you're growing cells in a petri dish, the bottom of your dish has to have some kind of, usually a polysaccharide, some sort of polymer on there that the cells can attach to so they can divide. In animal cells, the extracellular matrix of other cells frequently is the substratum they're attached to, and that allows them to divide. If they're floating around, they shouldn't be able to do that.